Okay, let's dive right into this masterclass explainer on ataxic disorders. Today, we are completely decoding the incredible clinical and genetic complexities found in Chapter 450 of the source text. If you've ever felt totally overwhelmed by the sheer number of spinocerebellar ataxias out there, or if you're just unsure of how to systematically approach a patient with gait instability, you are absolutely in the right place. We are going to break all of this down comprehensively.
We've got a lot of ground to cover today. We're going to look at approaching the ataxic patient, classifying ataxia by time, diving into the autosomal dominant SCAs, exploring late-onset ataxia, specifically SCA27B, and wrapping up with the autosomal recessive ataxias. So let's get right to it. Section 1. Approaching the ataxic patient. Let's start right at the bedside. Imagine a patient comes in. Their gait is unsteady, their speech is kind of slurred or scanning, and their hands are really uncoordinated. Where do you even start? Well, the absolutely crucial first step is realizing that not all imbalance comes from the cerebellum. We have to separate true cerebellar ataxia, which is pure imbalance with no vertigo at all, from its two biggest mimics. Mimic number 1 is vestibular disease. These patients will have a significant amount of dizziness or a spinning sensation, true vertigo. Mimic number 2 is sensory ataxia. In this scenario, the brain literally doesn't know where the limbs are in space because of a loss of proprioception. And we can test for this right there at the bedside with the classic Romberg sign. If the patient closes their eyes and their balance immediately falls apart, that's a sensory pathway issue, not a primary cerebellar one. Distinguishing these three right off the bat completely changes your diagnostic workup, and honestly, it saves the patient from a whole lot of unnecessary targeted cerebellar imaging. All right, Section 2, classifying ataxia by time. Once we're sure we're dealing with true cerebellar ataxia, time becomes our absolute best diagnostic ally. Think of this temporal grid as your ultimate diagnostic compass. It neatly separates out all the possibilities. So if you're looking at a sudden, acute focal event, something developing over hours or days, maybe throwing in some ipsilateral cranial nerve palsies, you really need to be thinking about vascular events, things like cerebellar ischemia or a hemorrhage. But if the ataxia is chronic, highly symmetric, and slowly progressing over months or years, the grid points us heavily toward genetic or metabolic origins. And then smack in the middle, you have the subacute symmetric presentations, evolving over weeks to months. This is exactly where you must aggressively hunt for toxic and metabolic causes like severe alcohol intoxication or vitamin deficiencies or even paraneoplastic syndromes driven by autoantibodies like anti-Yo or anti-Tre that are hiding in occult cancer.
Moving on to Section 3, the autosomal dominant SCAs. If that timeline points us to a chronic, progressive genetic cause, it is time to plunge into the genome. Take a look at this sagittal MRI of a 60-year-old man with SCA1. It's honestly striking. You can clearly see profound cerebellar atrophy. The beautiful, dense folia of a normal cerebellum have literally shrunk away. And that physical loss of tissue maps directly to the severe gait ataxia and dysarthria we see in the clinic. But what's actually causing this massive structural damage? To answer that, we have to zoom way down to the molecular level. For SCA1, and honestly a lot of these dominantly inherited SCAs, the culprit is something called a polyglutamine expansion. It's basically a genetic stutter. The DNA has these expanded CAG triplet repeats, and since the sequence CAT codes for the amino acid glutamine, this stutter produces proteins, called ataxins, that have abnormally long tracks of glutamine attached to them. Here's the key takeaway. These long chains create a toxic gain of function. The mutant proteins physically misfold. They clump up in the cell, completely gumming up the ubiquitin proteasome clearing system, which is basically the cell's trash disposal, and they literally choke the neurons until they trigger apoptosis, or programmed cell death. Now what's wild is that this exact same cellular pathology drives vastly different symptoms depending on the specific disease. For instance, SCA1, SCA2, and SCA3, which you might also know as Machado-Joseph disease, often show up with extrapyramidal signs, so you'll see rigid involuntary muscle movements right alongside the ataxia. Then you have SCA6, which is totally unique because the CAG stutter happens specifically in the CAXI-NA1A gene, the blueprint for a crucial voltage-dependent calcium channel. Or take SCA7, which completely stands apart because it features retinal macular degeneration. Those patients actually tend to notice blue-yellow color blindness way before they ever start stumbling. And of course, we can't forget SCA17, another major player. So it's the exact same root problem, but with wildly different clinical faces. But biology wouldn't be biology without a curveball, right? It's not always a CAG repeat, and it doesn't always make a toxic protein. Just look at SCA8. This ataxia is driven by a CTG repeat expansion, and it happens in an entirely untranslated region of the gene, meaning a mutant protein is never even made. Instead, it exerts its dominant toxic effects directly at the RNA level. It's a completely different mechanism of destruction, but it still relentlessly degrades the cerebellum.
Okay, Section 4, Late Onset Ataxia. Specifically, we're looking at SCA27B, which represents an incredible turning point in how we understand these disorders.
For decades, if a patient developed chronic ataxia in their 60s, they often just left the clinic without a genetic diagnosis. Traditional sequencing mostly focused on the exons, the protein-coding parts of our DNA. But by looking deeper into the non-coding regions, the dark matter of the DNA helix, researchers stumbled upon hidden intronic mutations that those older sequencing methods completely missed.
And that technological breakthrough led directly to the discovery of SCA27B. We now realize this is one of the most common late-onset inherited ataxias. It's driven by deep intronic GAA repeats in the FGF14 gene, usually presenting right around age 60. But here is the absolute game changer for you clinically. At disease onset, the symptoms are often episodic. And even more amazing, both those episodic symptoms and the baseline severity might actually improve if you treat them with a drug called 4-aminopreidine. We are finally finding actionable, treatable targets hiding out in the introns.
All right, let's bring it home with Section 5, the autosomal recessive ataxias. We're switching gears here to explore recessive metabolic and genetic breakdowns. And we absolutely have to start with the most common inherited ataxia of them all, Friedreich's ataxia. The pathology here follows a really devastating step-by-step cascade, usually starting before age 25. Step 1 is a massive expansion of GAA repeats, this time in the first intron of the frataxin gene, leading to a severe deficiency of the frataxin protein. Step 2, without enough frataxin, the cells develop profound mitochondrial iron dyshomeostasis. They basically can't regulate iron. Step 3, this massive energy failure and toxicity selectively wipes out sensory and motor pathways, causing the ataxia, absent reflexes, and high foot arches. And crucially, it also causes a severe life-limiting cardiomyopathy. Alongside Friedreich, we really need to talk about CANVAS. This is a recessive syndrome that's getting a lot more recognition lately. CANVAS stands for cerebellar ataxia, neuropathy, and vestibular areflexia syndrome. So if you see a patient with progressive imbalance, lost sensory nerve potentials, weird vestibular responses, and a chronic, unexplained dry cough, you should absolutely be thinking CANVAS. And just like SCA27B and Friedreich, it's driven by hidden genetic stutters, specifically biallelic AAGG intronic expansions in the RFC1 gene, which normally helps with DNA replication and repair.
All of this incredible molecular detail brings us to our final thought for this explainer. The landscape of these diseases is changing fast. We now have omaveloxolone, an FDA-approved targeted therapy that directly addresses the root mitochondrial dysfunction in Friedreich's ataxia. It's actually altering the course of the disease. So as we watch these granular molecular breakthroughs unfold, from untangling toxic polyglutamines to mapping out hidden intronic expansions, we really have to ask, how will these new targeted therapies reshape the future for patients facing these once untreatable neurodegenerative declines? Are we finally standing on the threshold of definitive genetic cures for all spinocerebellar degenerations? It is a fascinating time in neurology, and I really hope this explainer gave you the structured clarity you need to master it.